All right, welcome back to the Viking Initiative for Building Excellence. This is IB Biology, and today we're going to be talking about biostatistics. So, the first thing to know in biostatistics is how to construct a graph. You must know how to construct a graph on paper, and for your IO, you can do this in Google Sheets or Excel. There are 3 videos at the bottom of this page uh, that are great on how to do all 3 of these. Um, just a quick guideline though, to construct a graph, you will always need a descriptive title, x-axis and y-axis. You will need appropriate scaling according to the space of graph allotted. So this means that if you have a whole page, you want it to be bigger, and if you only have, I don't know, say just it's one FRQ and you only have a little bit of space, then you want to make it according. Um, you also want to use the appropriate type of graph. So this can be a scatter plot, line graph, bar graph, histogram, etc. And these all have different functions, so make sure you know what the right one to use is. And lastly, if required, you can use error bars to represent standard deviation if it's required for a mark on the paper. Okay, so we're going to talk about mean and standard deviation. So you should all be familiar with these two. Mean is just the measure of central tendency, or the average of all the values in your data set. You calculate it by dividing the sum of the values by the sample size. Standard deviation is the measure of the spread of a data. So if you look at these two symbols, sigma is the standard deviation of the population, and S is the standard deviation of the sample. And I've highlighted that one in yellow because that's the one that you will mostly use. It should be a bit smaller. 68% of the data lie within one standard deviation. You can see that in the graph. So where it says 0 at the peak of this curve is the mean, and left and right, 68, 34% each, that's within one standard deviation. And if you go 95%, that'll be 2 standard deviations, and 99.7% is 3 standard deviations. And as I said before, you can represent this graphically by using error bars. Okay, first test we're going to talk about out of 3 is the t-test. This is a statistical test to determine whether the difference between the means of two groups is significant. So we're going to talk about two concepts. One is the null hypothesis, and one is the alternative hypothesis. The null hypothesis states that there would be no significant difference. These are just two potential guesses as to what the outcome would be of the test. Conversely, the alternative would be that there is a significant difference. Another term is degrees of freedom. This is sort of accounting for the error within the test, and the calculation for this is the sample size minus 2 is the degrees of freedom. And you want to use the p-value of 0.05 so that you can be confident about your conclusion but still leave room for error. More on this later. Okay, so let's learn by an example. So a researcher weighed 8 Spalding basketballs and 8 Wilson basketballs in the table on the right. So if I were to apply the null hypothesis to this, I would say there is no difference between the means of both types of basketballs. The alternative would be there is a difference between the means. So I've calculated the mean of Spalding as 620 grams and the mean of Wilson as 580 grams. Okay, so there are two methods of doing the t-test. Method 1 uses the TI-84. So if you go to stat and then test, you will find number 4 is 2-samp t-test. So where it says input, you want to say data. List 1 and list 2 would be L1, L2. You'd have to type out those numbers that we saw on the table in these two. Frequency 1 and frequency 2 would just be 1 each. You would want to select the option that says mu1 does not equal mu2. This means that the means do not equal each other. That's what we want to test. Not that one is greater than or less than the other. This is something called a two-tailed test. This is not a pooled test, so you can say no, and then click on calculate. So you get the results that p equals 0.002. And with this p-value, if p is less than 0.05, because that was the value that we selected earlier, you want to reject the null hypothesis. That means that there is a significant difference between the mean weights of both basketballs. On the other hand, if p were greater than 0.05, you would accept, because and then you would say that there is not a significant difference. All this p-value is saying is that is what are the chances that this result happened because of some random chance or some likely chance. So if it's so small, it means that it would probably be a bit random if they were around the same. we can definitively say that it is not random, that there is a difference between the two means. So the smaller it is, the less random it is. Okay, so the method two, you're going to use the results from your TI-84. There should be a value that says T, and it will calculate that as 3.74. And we know that the sample size total is 16, so the degrees of freedom is 16 minus 2 is 14. So we want to find the critical value. You can see in this table here that along the top you have the different significance levels, and we want to select the one that says 0.05. We want to go down the degrees of freedom table to find 14 and find where they intersect. It will be 2.145. So if T is greater than CV, the critical value, which it is in this case, you also want to reject the null hypothesis. So both methods get us to the same conclusion. Okay, next is the chi-squared test. So this is a test to determine whether there is an association between two groups. So for example, in an IA, you could be testing, does the presence of one plant species, is it associated with the presence of another plant species, right? So the null hypothesis here would be that there is no association. The alternative would be that there is an association. The degrees of freedom is calculated differently here. In your table, it would be the number of rows minus 1 times the number of columns minus 1. And another number you need to know is the expected value. And this is calculated by the row total times the column total divided by the total. So we'll get to that now. Okay, so let's look at another example. A researcher wants to determine whether there is an association between the size and price of a house. Logically, there is an association, so let's see what the results are. The null hypothesis would that be there is no association between the size and price, and the alternative would be that there is an association between the size and price. So we're going to make something called our contingency table. And on the left-hand side, you're going to have size, and along the top, you're going to have price. And so, for example, in the price range of 250,000 to 500,000 and a size of 1,000 square feet, there are 30 houses. And the same goes for 20 for the other 1,000 square foot house and 20 and 30 for the two 5,000 square feet categories. So the total of each row is 50, the total of each column is 50, and the total of both the rows and the columns is 100. So now we have to construct our expected values table. So the way we do this is row total times column total divided by total, right? So that would be 50 times 50 divided by 100, which is 25. That is the expected value in each of these rows, each of these cells. Okay. So first off, the method one is TI-84. If you go stat test and you find C, you will find x squared test. This is chi-squared. Where it says observed, you want to find the matrix A. You will have to go to this matrix and edit it to match up with your expected values table, with your contingency table. And then where it says expected, you can leave that matrix B blank. It will autofill it based on what you put in your observed values table. Then you click on calculate, and the results show that P equals 0.0455, meaning that you will reject the null hypothesis because it is less than 0.05, meaning that there is a difference between, there is a significant association between the size and price of a house, not a difference. And that is what we would expect. Okay, so in method two, this could be using a table. So you would find the chi-squared number to be equal to 4. The degrees of freedom for this is 1 because there were 2 rows and 2 columns. So 2 minus 1 times 2 minus 1 is 1. And if you find 0.05 and you match it up with the degrees of freedom of 1, you'll find a value of 3.841. And as we said before, if the test statistic is greater than the critical value, then you reject the null hypothesis. So this is the same conclusion as we got to last time. Last test we're going to do is called ANOVA. This is analysis of variance. And this statistical test is to determine whether there is a difference between the means of 3 or more groups. Okay, so this is similar to a t-test, except it is between 3 or more groups. And this is likely what you will use more often because t-test is quite limiting. Okay, so the null and alternative hypotheses will be the same. So we're going to go back to the same researcher from question one, and now they've added Baden basketballs into the mix. So here's the table of all three basketballs. So if you go back to the TI-84, this is really the only method for this. You'll find letter H all the way at the bottom of the statistic test, and you will click on ANOVA. For the TI-84, just normal edition, not color edition, it'll open up in the normal window with ANOVA and a parentheses. And here you want to put in L1, L2, L3, or however many lists you have. In this case, we have 3 lists because we're comparing 3 types of basketballs. In color edition, it may be a bit different, but this is how it is in the normal edition. So in L3, L1 and L2 are already filled from the previous problem. L3, you go ahead and add the Baden basketballs. So your results here, you get an F ratio equal to 7. This is sort of what it's like to get the x squared or chi-squared value in the chi-squared test, or the t value in the t-test, right? So the first thing we can look at is that p equals 0.00468. So p is less than 0.05, so we will reject the null hypothesis, meaning that there is a difference between the mean weights of these 3 basketballs. Alternatively, we could say that if f is far from 1, which it is, it's 7, you also reject the null hypothesis. So with each of these tests, there's 2 methods that you could use to get to the same conclusion. With ANOVA, if you're not using the TI-84, chances are you will get the F ratio or the p-value, so you'll be able to make the conclusion on the test. You will not have to do it by hand. Okay, so AP Statistics covers the t-test and the chi-square test, so they're more likely to go into a little deeper than you would need for bio. But if you want more clarification on these 2 tests, check out those resources for more help. For ANOVA, the extent of what I've covered is pretty much all you need to know, so that should be okay. And yeah, that's all it. Just normal reminders. Check out the website for more resources. If you have any questions, again, you can join the group chat and ask us. Feel free to. And yeah, thank you for watching.